Okay, so welcome to Palacký University to the Advanced in Silico Drug Design workshop and / hackathon. My name is Karel Berka and I'm the main organizer of this event. I'd like to welcome you specifically to this place, as this is the Faculty of Science, and we are doing this workshop together with collaboration with our colleagues from Institute of Organic Chemistry and Biochemistry of Academy of Sciences of Czech Republic, Uppsala University, and University of Vienna. And the payment for the lectures and for the space is provided by Elixir Infrastructure in the Czech Republic. So, I'd like to first stress that the drug design nowadays, especially the in silico drug design, is like a tool. And it's a similar tool for the designing of new compounds as the NMR is. Because you need NMR, but it don't solve every of your problems, but it will give you some clues. Similarly, in silico drug design will give you some clues about what to do with the drugs and how to provide them further. Disclaimer is, I haven't yet designed any drug in a common use, and since the design of the drugs is much longer process, I think that Professor Thierry Langer from Vienna has some knowledge about this. So, what shall we do? Today, it's basically we will be speaking about basics. Tomorrow it will be the whole day informed for the ligand-based methods, where we start with QSAR modeling, and then we'll switch to deep learning. And in the afternoon, we will switch rooms for the computer room, just along the hall. And there we will have 2 tutorials, one on the Python, one for the deep learning. On Wednesday, we will be switching rather towards the usage. So, oh Jesus, that's wrong. Sorry. There is a lecture about de novo design from Pavel, and then there is no tutorial in the morning. There is a drug delivery by in silico by Per Larsen. Then, in the afternoon, there will be a lecture about the pharmacophores and tutorial how to use them. And then there will be a workshop dinner. Whereas this year, we don't have a sponsorship for this dinner, but we will provide the space for you so we can convene together and go and speak in more informal way, even though I hope that this event as a whole will be informal enough. I forgot to mention the excursion, which is tomorrow, and that's done by Pavel, who will show you, those of you who want to go to the IMTM, the institute at the Faculty of Medicine, where there is some devices about translational medicine. On Thursday, it will be on me, and it will be basically structure-based methods, and we will be speaking about the molecular mechanics, modeling in general, and docking. And in the afternoon, there will be 2 tutorials on that. And on Friday, there is a special time. Ve třídě. Teda u mě v kanclu, anebo uhlek. On Friday, there will be a challenge where Pavel will select some interesting problem for solving to find out the interesting ligands, and we will score them. And this will be up to you to use the knowledge you have already or the knowledge you will learn here. So, do you have any questions about the program? No? Cool. Let's go. Yeah, that's it. I'll be speaking about the stuff, but it's of course important to think about the future on the topic. And one book which I found really nice for the entrance on the field is the Dave Young Computational Drug Design. This book is really a nice interview into what types of the individual, what different methods can do for you. Unfortunately, it's relatively old, even though it's just 10 years. And there is not enough coverage of the machine learning stuff, which starts to be the thing in the field last 3 or 4 years. So, and one thing which I really love to read is the In the Pipeline blog from Derek Love at the Science, and that's really a nice feature about what is going on in the drug development in general. So, stating that, Misha is now writing the password for UP-guest Wi-Fi. Isn't it UP-guest? UPOL-guest? Nemenuje se to. I think it's UPOL-guest Wi-Fi. With, no, normálně s pomlčkou. So this is the password. If you don't have EduRoam, which is also available here, this is a possibility to connect your devices. So I think that the introduction is enough, and now I should speak more about where do we get the drugs. So the oldest drug, as a typical one compound, is taken this compound, morphine, which was developed in 1806. And this was used almost everywhere because it was a nice painkiller. A little bit addictive, yes, but it was used for everything. Like it was used for a small children to take a sleep with this morphine drug, so everything was completely okay. And this even shows you several things already. The source of this morphine drug was from plant. So quite a lot of drugs nowadays is still coming from the plant or plant derived from the plants. It's a relatively complex molecule, even this. Still relatively small, and small molecules are still making the majority of the drugs we still have in use. And you have different ways how you can deliver it. Okay, you can have ointment, you can have some syrups. These drug delivery options are also quite changing the way how your drug will be perceived afterwards by the organism. And it already showed that you can have some side effects, which can be quite problematic, and therefore several drugs had to be removed during the so their derivatives, which are more safe, can be defined. So we have all of this in this drug, and basically when you seal it, you start from the plants, then you go to the extractions, then you go to the direct chemicals, then you go to the something coming from the synthetic chemistry or pigments like colors. And then you go nowadays for high throughput libraries where you mix everything together and you have thousands or millions of compounds already. It's interesting that we also changed how we test the compounds, because for a long time we were testing basically on humans, and only in late years it was on humans/prisoners, and only afterwards it started to be a little bit unethical to try the drug immediately on a person, so it was tested on animals, then on isolated organs. Nowadays it's on cell lines. Okay. And before that we have into in silico, so we can go into virtual screening already. So everything a little bit changed over the years, but still the amount of drugs which is coming is that you have relatively large synthetics nowadays, but if you count for the natural compounds or natural compounds derivatized on synthetic but mimicking natural, it's a relatively large amount as well. And you have like 14% of biologicals, which is like the vaccines and stuff like that. The idealized drug design project is that you come with some kind of illness, you isolate the cause, then once you have the cause, like specific target, then you start to develop the compound that can be attacking you to this target. Then you go for some preclinical testing on animals, and then you have to formulate it so it will be working. Then you file IND application for either the FDA or we are in Europe, so a European Medicines Agency. Then you have human clinical trials, and then you, if you are extremely lucky. Because just from the number of compounds which are entering the formulation after the formulation stage, only 1 of 10 is the one which succeeds. The 9 usually are taken out from the clinical trials. So, I was already speaking about some targets, lead, hit, and so on. So let's define them. So the target is the biomolecule which is interacting with the drug. So that's the protein onto which we are targeting. Lead is a base structural motif that gives you the general idea about how the drug should look like, and you can afterwards develop it, so you can derivatize it somehow. Hit is just a compound which, from the series of compounds you've tested, you usually select some kind of a criteria, like the compounds which have the activity of range of nanomolar. That's probably too rough, so low micromolar. And those compounds which pass through that through that screening are the hits. So these are the compounds which generate the candidate compounds which you are moving towards the next phase of the drug development. And then you are specifically interested into efficacy, which is does quality works or not, or the activity, which is a quantitative property about how much does it works. So how big the dosage needed to be. The completely best is that if the drug is active even at the picomolar concentrations, because then if you scale up into a whole human, then you are in a range of milligrams. That's cool. If you are at nanomolar, then you are about the tens of to hundreds of milligrams. If you are above, like micromolar, then this can go into almost grams. And that was the trouble with the HIV cures before the Antonin Hollies, antivirals, because they were not sufficient enough for the people who from HIV had to eat like 10 grams of medicines every day. It does not seem to be much, but it's still a relatively large pill. Yeah, and if you are at millimolar, then, well, forget it. If we are speaking about a target, we also have to speak about how the drug is able to go to the target. So to speak about the bioavailability, whether the drug will be able to go there in a real case, in necessary concentration. So that was how we develop the drugs and how much do we actually develop. I guess you know the Moore's law, because you are working with computers, so you know that every 2 years the number of CPUs doubles. The number of processors on CPU doubles. And therefore the speed also doubles roughly twice. The problem in the pharma industry is that we have a different way, and that the money that has to be spent on drug development is doubling over a similar time. So when you take how much of the drugs you can develop by the billion of US dollars, you can see that there is a relatively not nice scale, logarithmic, that gives you that in the 50s, 1 billion US dollars, which were leveled to the today's US dollar, was actually, you could develop 100 drugs. Nowadays, for the same amount, you are below 1. And the last number I was able to find when I was updating this lecture from the last year was something like $2.5 billion US for development of one compound on one drug. And the majority of the troubles here is that you halve per 9 years the efficiency of the drug pharma industry. Even though since the 90s we have the biotechnologies which can help you to build the new molecules efficiently, and you have also the in silico drug design, and still we are still going down. So the trouble is that even though there is plenty of development in the field, it's still more and more expensive to develop a new drug. It's different whether you are looking at the overall trend or just the rate of decline or the adjusting for 5-year delay on spending impact. All the time we are going down. And there are several reasons for this. There is first problem is better than the Beatles. Because when you are developing a new drug and you are developing against the drugs which are already at the market, you have to be better, of course. But if the drug is efficient enough, then developing additional drug to this let's say, illness might not be good enough to pay for your development. So that will increase the price for those drugs. And especially nowadays, when we have several large, big pharma, and they are trying to get the drugs for everything, almost. Therefore, they are developing into their own medicine for the illness which already their competitors have. And so we are losing money on that. Then there is another problem, and that's the problem of the cashless regulator. Because if you are developing the drug, of course you are messing with the organism, and therefore it is necessary to look for the toxicity possibilities of the new compound. And this toxicity testing takes quite a lot of time, and it's also quite extensive. So that will increase the price again. Then there is a tendency of the investors just throw money in it, just give more money and more money, so it will give. The problem usually is not that you don't have enough money in the pharma industry. The problem is that you don't have enough of the ideas. That almost everyone is shooting for the same targets. Okay? And developing against the same targets basically means that there will be competition and therefore quite a lot of lost money on it. And the trouble is that the basic research is not that easy as it looks like. So it's the biggest trouble which we have is that sometimes we don't understand the illnesses. Typical example of this is Alzheimer's disease. The previous generation of anti-Alzheimer's disease cures were looking into alpha-beta, how was it called? Prion-like peptides, which are forming those filaments in brain of people with Alzheimer's. Therefore, the first drugs were trying to target this type of the filaments. Then it was found that the filaments by themselves are not problematic, they are not toxic. But probably the small oligopeptides, which we don't see, might be causing the trouble. New round of the cures were developed for this, and it was not active again, because now it starts to see that this might be just a side cause of something else. So now there is another possibility that it might be a tau protein that is causing the trouble. Okay? And we don't know yet whether this tau protein is the one or not. So that's how the field can move along the illnesses, that you basically have to find out the real cause, and it's really hard to get the real cause right. And of course, if you are developing against the wrong target, the cure won't work, of course, at the end. Yeah, so the problem is that the human body is not that easy as the car, I would say. However, last 2 years were relatively successful in how much of the new molecular entities occurred and were satisfactorily approved. So I've looked into the last numbers of the FDA, and last year the FDA registered 59 new drugs, and there is a steep increase from the year 2016. It was one of the worst in the decade, and basically there is a relatively nice optimism in the field that the number will grow even though, because now the drug development does not go into a one drug for all model, which used to be in the 50s, 60s, and so on. But nowadays, there is also a possibility to develop the drugs for so-called orphan diseases. Those are the diseases which only one person in thousands can have in a population. You would think that this is so small number that it would not have any possibility for development of new drugs for such kind of relatively really rare diseases. But there is an interesting way that the orphan diseases need not to have so much of the toxicity testing, because there will be much less people who would be needed them. And therefore, the toxicity screens are not that strong as when you are developing against all of the drugs which will go into full populace like ibuprofen. Yeah, so it is interesting that these rare diseases now makes approximately 50% or 40% in case of Europe. Yeah. And that's, since this is easier, this allows small and medium companies to develop their own drugs for the rare diseases. And it starts to be really a thing, because if you think that 50% of this number is just the rare diseases, it gives you quite a lot of boost. There was also a speaking in the literature that there will be more and more biological cures. However, the number of biologicals is relatively stable along the last decade. And it's making like 20% of the drugs which are registered. In Europe, we have more biological drugs developed than the small molecules, which is an interesting difference between the Europe and United States. And yeah, so unfortunately we can't see much, but you will see afterwards, because the slides will be on the web of the course. So this is where we can get the drugs and how we can get the drugs to be approved in a really short way. And now let's switch into how we are working with the drug design. So I've already said that it's an expensive problem. Okay? This is the number from the Tufts Center for Study of Drug Development from 2014. And that really accounts for quite a lot of bad development which was done along the drug which was successful at the end. Because you have to think about the development costs for not only for the development itself, but also for the production afterwards, patents, distribution, and so on. The trouble is that afterwards, the new drugs are quite expensive, and it can be over 1,000 per dose of such drug. It can be even more. Yeah. And not only expensive, but it's also a hard problem, because the identification of target drug pair is not really simple. You have to think about the bioavailability of the compounds. You have to think about whether the drug will be able to get inside the body, so absorb, whether it will be able to distribute through the body, so the D, whether it will be metabolized, how it will be eliminated from the body, and then about the possible toxicity effects. So this is this ADMET effect. And you have to think about possible side effects, because usually if you are developing a small compound, the small compound can bind not only to your target, but it can, to some hopefully lower level, it can bind to another target. A typical problem is when you develop the inhibitors for kinases, because there is a large family of protein kinases, and most of the inhibitors are actually binding to the sites where the ATP binds. But ATP will be taken by quite a lot of those kinases, so there is quite a lot of counter crosstalk that your inhibitor will not only be inhibiting, let's say, CDK2, but it will also take CDK5, CDK9, and others. So then it's much harder to get really a selective targeting. The question is whether you actually need the selective targeting, whether sometimes it can be helpful that you are not going to one spot, but that you are blocking more kinases in the total. And I said that it's expensive. So let's say that the cost per one compound in virtual screening, let's say, when we're speaking about the electricity, the personnel, the computer hardware, and so on, let's say 100 Czech crowns. If you run the biochemical analysis, it's 700 Czech crowns, and Czech crown is approximately, let's say, 25 Czech crowns, €1. So divide by 25 and you get it in euros. Testing in cell cultures is more expensive. Then if you add it on mice, then it's even more expensive. If you want to get the structure of your protein target, and quite expensive experiments. Then if you want to do the efficiency testing, then you have to specifically get the animal that have the disease, or somehow, so you have to induce the disease in the mice, let's say, and then you have to cure it. So that's of course much more expensive because you need to do the gene modifications and stuff. If you want to do the chronic toxicity, then you have to have those animals for longer time period, and you have to have a cohort of them large enough to basically be able to get through all of the effects along the lifetime. So it's more expensive as well. But it's everything like that is nothing in comparison to the clinical testing on volunteers. Because in case of volunteers, you have 3 different phases. So first, you test the security, let's say, that whether it's safe to put that. There are occasions like 2 or 3 years ago, they had in France, they had the compound which was okay on animals. They put it into a small group of 10 people, and out of those 10 people, I think 4 died, and the rest had some severe disease afterwards from that, just from this testing. So even though we have the testing of toxicity on animals beforehand, we are not completely sure that it will not be toxic on humans, because we are different organisms, of course. Then after the safest, then you want to look into the efficiency. So you are testing the dosage, which will be good enough to cure, but low enough not to have the side effects. And then the last step is the clinical trial of the large cohort of the population. So you take the people from all around the world, because the different mutations along the people might affect, of course, the toxicity of your compound again. Yeah? And this is the most important. This is most expensive, because that's the largest study you need to do. And even afterwards, once you get your drug approved, there is a pharmacovigilance afterwards that you afterwards receive, or the FDA, or in Czech case, SÚKL, Státní úřad pro kontrolu léčiv, or EMA, European Medicine Agency, are still looking into the drug and trying to get whether there are some reported side effects from the drug from the pharmacies. Yeah. So even afterwards, after some usage, it can be changed that there can be some kind of labels. Don't use this on pregnant women, or don't use it together with the ibuprofen, or whatever, something like that. I've already spoken that it's a hard problem, but the trouble is that we don't know. Sometimes we basically don't know what is the cause. I've already spoken about the Alzheimer, and if you speak about it, we have something like 20,000 genes. That's gene in a definition of the protein, the part of the DNA which afterwards make the protein. But we have also quite a large role of RNA nowadays, and that's a relatively large field which is still developing, so we still don't know how much of these RNAs are important for what. So we still don't know how much this thing will take care, and slowly the development against the RNA starts to start up. And of course, there is quite a lot of interactions between the proteins. So sometimes you develop the drug not only to target to one specific drug, but you want to target the association of two protein units. That's something that would disappear, and that's also something, a new thing, because to get the right protein structure is really hard. Usually it's something like 2 to 10 years when you have some kind of lead molecule that is active, that towards the clinical testing. Because for the application, for the drug approval, you need to know how it works. Yeah. And you need to specify it. And that's one of the reasons, as I've said, one successful out of 10 drug development projects. And the major problem nowadays is that the compound we have is usually safe, but it does not have activity. So that's the biggest trouble, that once we get the compounds really safe, we get compounds which are not that active. And sometimes the placebo is more active than those drugs afterwards. There are multiple problems which can go along the project. So the first problem is non-existing testing model. Let's speak about HIV. HIV, the H, is a human immunodeficiency virus. Therefore, to test it on mice, it does not work because the mice does not have a human immunity. So there was a large amount of humanized mice that should have the human immunity, but so far as I know, nothing worked. Yeah, so in that case, in HIV, you had to go at least to the apes, and with the apes, it's much more troubling to test on them. So there is also a problem, you can't test on the people immediately. You have to have at least the toxicity screen, so you think that at least the drugs to some extent will not kill them quicker than their illness. The rare diseases, as I've said, orphan diseases, have another trouble. If you would have the rare disease, the development in one country will not be enough, because in that one country, you won't have enough of the people to test on. So, for instance, in Europe, the Elixir has one of the group for rare diseases that is covering all of the Europe and trying to connect all of the people with different rare diseases along the whole Europe. So, at the end, there will be enough of people to test the drugs on and test the drugs for as well. So that's afterwards the orphan drugs have much smaller amount of how much money can be get back from their sales, because there wouldn't be so much people who would be buying it. Yeah, so the trouble is that they would have higher prices. Yeah, toxicity, and yeah, I've already talked about this. The trouble is also with the illness type. So what do you want to do? In some cancer, you can have that some kind of enzyme is overproduced, typically the kinases. Then you can develop the inhibitors. Right? And that's one of the, I would say, relatively easy way, because to find good inhibitor, you need to find a compound which is binding stronger than the natural ligand. And once the compound is binding stronger than the natural ligand, then you lower down the enzyme effect, and so on. Sometimes the trouble is the high response from receptor. Similar, if you have high response from the receptor, so in this case COX in pain, you can use pain relievers, which will be basically antagonists of this signal. They will again go and bind towards the receptor, and again they will bind strongly, so it's similar to inhibition. The trouble is that sometimes you not only have to develop the compound which will bind, but which will also activate the receptor. And that's when you have low response of receptor. So for instance, you have a low amount of the serotonin, and therefore you want to cure depression, for instance. And one part of the depression can be solved, can be say, as that you have low amount of the serotonin in your brain, and therefore you need to increase the response of the receptor towards that small amount of the serotonin which you have there, and therefore you have to find out some kind of agonists which will activate the receptor. That's far more harder, because in that case you are not only looking for the binding, but you have to look also into the action, and that's much harder. There can be a regulation peptide. So for instance, this CGRP peptide in migraine can bind to some kind of antibodies, okay? And that's leaving us into biologicals, which can be much more specific than the small molecule are. And in RNA, you can do quite a lot of things with the RNAi, so inhibitions of RNA production, some aptamers, CRISPR, plenty of things nowadays in this field. So here we will be speaking basically about the small ligand with protein. So we won't go into those things. And in that case, we basically are working in the idea of lock and key analogon from the Nobel laureate Emil Fischer, where we have a small molecule as a key and large molecule protein as a lock. So which locks do we have? So how we can find the drug targets? This is the analysis of the molecular drug targets, which was published 2 years ago already now. Well, I think that those 59 new compounds does not change the scale so much. And you can see how much of the compounds is actually percentage of total of the number of drugs. And the large number of the drugs targets, which we are now using again, is basically GPCR. That's a G protein connected receptor. And this is basically a sensoric type of the protein, which is in a plasma membrane, that takes the chemical signals from above, from extracellular system, and then transfer it into a signaling pathway inside the body, inside the cell. So since you are looking into a signal, then it's much easier to get afterwards the response from the cell, and therefore to have the effect you want to have. Another thing is again in the membrane, so it's ion channels. Those are, for instance, the compounds which are working for the heart, because in heart you have the efficiency, the work cycle of the heart is organized by a specific opening and closing of ion channels. So this is the quite a lot of important things there. Then there is some kind of kinases, well, 10%. This is how much of compounds are active. And then you have nuclear receptors. So again, it's quite kind of a receptor. And then you have all of the others. But majority is basically these GPCRs and ion channels, and that's connected with the how much of the compounds is actually a drug, and 30% of the drugs is developed against the GPCRs. There are some cases where the percentage of compounds which are in a Campbell database, which is a database running by the European Molecular Biology Lab, where they collected all of the compounds from the patents, from the journals, and so on. So they get quite a large collection of the real molecules which were at some point tested against something. And you can see that these two numbers basically match. So the number of compounds which are in the database and the number of actual drugs are basically the same. I was speaking about the kinases. And here you can have much different view, because there is a much more of the kinase drug development, but not much of the compounds at the end. Yeah. The same thing is for the protease, basically HIV. Yeah. In those cases, you have much more of the development going on than the actual drugs. There are, of course, different areas, so these nuclear receptors and ion channels have much less development compared to the end of the real drugs at the end. So sometimes it's interesting to look into which field is overly making the claims, like the kinases, and which field actually makes the drugs at the end. If we look into the ATC code, into the classification of the therapy according to the anatomy, what are the pharmacological action, subgroup and subgroup, then we can select about which ATC codes have their own drugs or the small molecules or biologics. And we can see that there are some places which have a relatively large number of compounds, like nervous system, and some places doesn't have, like the blood and blood-forming organs. So this is not completely the same, and if we look into, again, into individual groups and how much of the groups have the development there, again, against the approval rate, we can see that in the last years, we have quite a lot of immunosuppressants and antineoplastic agents, but not much anything else. Quite a lot of drugs was there even before 1990. So most of the drugs basically we know from the past, and only certain areas have seen some development during the last years. So again, this might serve as a nice clue about where to go for your drug development, to go for something new. Yeah. Another thing is that when you look into Campbell and you analyze where are those drug targets actually located, you can find out that most of the compounds is actually membrane receptor or enzyme which is located into the membrane, and some ion channels as well. So basically more than 60% of the drug targets nowadays are basically some kind of the membrane-bound proteins. Basically the major reason here is that once the signal is coming from outside, then the membrane is the first envelope around the cell, and therefore the drug does not need to go inside to get some kind of effect, but it just needs to go on the side, on the membrane, to get the effect afterwards. So it's much more easy to get the drug action from the receptors, basically. Yeah. I think I'm starting to be a little bit long. Yeah. And therefore, I'll speak about this what we have as a possibilities. Depends on how much of the knowledge we already have about the system. Because if we know the ligand and we know the structure of the target, then we can use this knowledge for the structure-based drug design, and we can use, for instance, docking to do the analysis. If we know the ligand, but we don't know the structure, we have to use ligand-based drug design. And depending on how much of the compounds we have, the active ones, we can look into a different type of the methods, which will be shown mainly by Pavel and Thomas tomorrow. If we don't know the ligand, but we know the structure or the target, we can look for the de novo design, so basically to develop, to find out what might possibly be the active compounds according to the structure of the target. And if we don't know the ligand and we don't know the structure, we still have a possibility that we can look for the compounds which might be bioavailable, at least. Okay. So before any testing, you can select those compounds which might have some potential in them. For biologicals, even though we won't be speaking about the biologicals for the rest of the course, but I want to show what are those compounds which are taking like 20 or 30% of the drug approvals nowadays. It's some kind of biological product. It's different in the US and European Union, and that's probably the reason why European Union has more of those biologicals approved, because their definition is basically larger. And in the US, it's a biological product where any virus, therapeutic serum, toxin, antitoxin, and whatever used for treatment of cures and diseases or injuries of man. And here we have also a diagnostic properties, therapeutic properties, and some kind of direct extraction, and not only the direct, but also the difference. The biologicals have trouble with the production afterwards, because chemicals, medicines are basically chemicals, and once you have the recipe and you follow the recipe, you get all the time the same amount of the drug. While the biologics are grown, and therefore you have to be much more careful about the ways how you produce them. Yeah. For instance, there is a quite a large collaboration between this faculty and pharma company Teva in Opava, in northern part of Czech Republic. And they had the trouble that they are working on immunosuppressants. But the trouble is that they have the, I think they are working on paclitaxel. And the amount of the compound which is produced by the cells is lowering during the years, because the cell starts to be resistant for the compound, and therefore they had to produce much larger amounts of the drug producing cells in order to get the same amount of the drugs at the end, and which is of course wasting of the resources and so on. So they really need to rework again how they produce this compound. And you have to purify the stuff and so on. So it's much harder. The problem is that the biologicals are much more complex than the small molecules as well. So you have also a different variety of different resources from where you're taking it, like from the blood or blood derivatives or individual blood components. It can be the production of the proteins itself, like immunoglobulins. It can be some kind of human tissues themselves. Some xenotransplantations, right? Something grown on pig and transferred to human. There can be some kind of gene therapies as well. You probably heard about the CRISPR babies, which should not get the HIV. And also some vaccines and anti-allergic extracts as well. So there is a large amount of different biological products which can be produced, and their quality assessment is much harder. From us, like in silico, they are also quite different in size. So this is aspirin, like the size of the small compound up to 500 Daltons. And this is monoclonal antibody, which is 100,000 Daltons. So it's much harder to develop something on this scale than on this scale in silico. And of course, again, there is another trouble that the protein you develop then undergoes some kind of post-translational modifications, which will again change how the compound works and how the whole system, how the whole protein can be affected. So it's much harder to make the rational design on that. So I come to the end of my lecture. So some take-home message from this. I hope to know you that the drugs comes from various resources. I've shown you that still majority of the drugs are coming from the derivatization of some natural product. It's hard and expensive problem, mainly due to the costs of the clinical trials and biology behind. And the most typical drug targets are GPCRs and channels, NUS receptors and kinases. But there is a long tail of orphan drug targets. And I've shown you that those orphans can actually give you the drug much more easily at the beginning. So it's possible to go and look into that specific orphan and to help it with it. And the biologicals are much more complex, and the development for them is, of course, much more complex. And unfortunately, there is no gold path for the drug development project, because all the time you have to tune the project according to what you have, what are the knowledge, what are compounds, and what you want to achieve. And that's all from me.